Welcome, science friends, to movement across membranes, or transport. So the first two slides we're going to see are a sort of recap of our last video, just to refresh our brains about what proteins are when we're talking about cell membranes. Protein channels are doors that go into and out of the membrane that help very specific molecules through the membrane if they can't fit through the phospholipid bilayer. Mostly that's larger molecules or things that are ionic or have some sort of charge and need a little extra help getting through the membrane. It's a lot like fitting a square peg into a round hole if it's not the right molecule. So just like how enzymes have specific substrates, same thing here, these proteins can only accept specific molecules. There's special proteins for amino acids and special proteins for sugars and water and all sorts of different things that need to come into and out of the cell. So movement through that channel, that protein channel. Why do molecules even need to go through the membrane if you give them a channel? Well, we're looking at what we call concentration gradient. There's always going to be more of a molecule on one side of the membrane than the other side. So we have a higher concentration versus a lower concentration of that molecule. When molecules move from high concentration to low concentration, this is natural. This is what happens in most things. And when we're talking about membranes, this is called diffusion. We move naturally from high concentration to a low concentration. And in our picture here, we can see that if we're looking at just molecules of dye in water and we have some sort of semi-permeable membrane, as time moves, so does our dye. And it goes from that high concentration on one side and it slowly goes through the membrane to even out that concentration. Eventually, even amounts of that dye, or whatever molecule we're looking at, is what we call equilibrium. It's the same on both sides. But until we get there, things always go from high to low. Here are a couple of GIFs that are showing us what we call passive transport. Now, since we're moving with the concentration gradient from high to low, this means we don't need any extra energy. It just happens naturally. So in our first little GIF here, we're seeing red dots moving out of this membrane. It's very concentrated on the inside, and naturally it wants to even out those concentrations between the inside and outside of that cell. So it's naturally going from high concentration to low concentration. In our other GIF here, we're seeing the diffusion of water. We can see those green molecules there. We're also seeing water molecules, those the blue ones with the two little hydrogen atoms on there, and we can see them, they can move straight through that membrane. No energy required. There's more water on one side than the other side, so as it's passing through the membrane, it's trying to reach that equilibrium of same amounts of water molecules on both sides. We can also see in this that the green molecules are too big to fit through the membrane. They would need a protein channel to help them pass through to the other side to even out that concentration. Once again, I really want to stress this vocab, concentration gradient. That's the difference in concentration of molecules across a space. And in our examples that we're learning about, that space is the inside and outside of a cell. Always want to move high to low. It's very natural to go high to low. So the concentration gradient is how many molecules of one thing are on each side of something like a membrane. We just talked about simple diffusion. Really want to touch on it again. This is high to low concentration, goes straight through the membrane between those phospholipids. And so some of the substances that move with simple diffusion are things like oxygen and carbon dioxide, water, fats, alcohol. These are all things that can slip between the phospholipids of the membrane. Now in this example, we have fat. Looking at the different concentrations of the inside and outside of the cell, we see that there are a lot more fat molecules inside the cell than the outside of the cell. And you can also take note of the concentration high inside and low outside. So if we're working with simple diffusion and our molecules always want to move from high concentration to low concentration, which way will the fat move? It will pass through the membrane from the inside to the outside because there's more fat inside and it wants to move from that high fat concentration inside to the low fat concentration on the outside. The next type of diffusion, again, diffusion is no energy, just going from high to low. Facilitated diffusion means these molecules need help. They need facilitating. And what that means is it's basically the same as simple diffusion, except this time it needs a protein channel to help. Something like sugar, it's a large molecule. And when it's a large molecule, it needs a little extra help. It can't just slip through the membrane. So, for example, sugar, it needs that facilitated diffusion going from high concentration inside the cell to the lower concentration outside the cell. It's going to move from inside to outside because we want to even out those concentrations. Other substances that move through facilitated diffusion are things like carbohydrates, like sugars, amino acids are fairly large, nucleotides, and ions. Those are all great examples of things that need the protein channel to help get them through the membrane. Once again, diffusion is from high to low. It can either be simple diffusion that goes straight through the membrane between those phospholipids. Because it's going from high to low concentration, we don't need any energy. And there's also facilitated diffusion. It needs the help of the protein channel. It's too big to fit between the phospholipids, so it needs that doorway to go through from high concentration to low concentration, whether that's into the cell or out of the cell. The last type of transport is called active transport. Now, sometimes cells need molecules to move against the concentration gradient. That means it's going against what's natural. Instead of going high to low, active transport goes from low to high. And because it's going against nature, we need to add energy. And of course, energy for all of our biological organisms is ATP. So our protein here is actually pumping against the concentration gradient, or sort of uphill, and uses that ATP energy to go against the concentration gradient. In our little GIF here, we're seeing how the energy is helping our protein go from low concentration to high concentration. We're pumping against that gradient. So as a quick little sum up here, simple diffusion, high to low. You can see we have more of those little purple circles on one side, and it's going into the side with fewer of those circles, straight through the phospholipid bilayer. No extra special treatment needed. Then we have facilitated diffusion. Again, this is passive because we're going with the concentration gradient from high to low. There are more triangles on one side than the other, and we're going in that direction from more triangles to fewer triangles. And lastly, we have active transport. This is the one where we need energy, or ATP, to go against the concentration gradient. As you can see in the image here, we're going from fewer to more. So again, it's going from low to high, which means we need energy, or ATP. This is just a little cheat sheet that is helpful, kind of identifying all of the different things that are happening, small and nonpolar things, large molecules, ions, hydrophilic molecules. Water is a special case. And anything going against that gradient is going to be active transport. The last little bit here, exocytosis and endocytosis. These are a couple of those words that sound similar, but because of that very slight difference in their prefix, mean opposite things. So exo means outside, so exocytosis, cytosis means cell, means that we are moving things out, outside of the cell. And we're using exocytosis to move a lot of stuff at once. It's a bulk transport. Endocytosis, endo means inside. We're moving lots of things at the same time, or a bulk transport into the cell. Now, because we're moving a lot of things, this requires energy, so it's considered an active transport. Here are some images that help us see the difference between exocytosis and endocytosis. In the first panel here, we see exocytosis. We have a little tiny bubble. It's actually a vesicle. Its outside is made up of those phospholipid bilayers, just like our membrane, and that bulk transport, or a lot of those little tiny purple circles, need to exit the cell. So it's moving to be released by the membrane. The second panel here is endocytosis, where we're just seeing the opposite happen. We're moving a lot of those green circles from the outside of the cell to the inside of the cell, and we're doing a lot of them at once, and we're just pinching off a piece of our membrane to create that vesicle or that little bubble. Here's just another explanation, another image of the endo and exocytosis moving in versus moving out. And lastly, our little sum up page here. Two things that require energy are active transport going against the concentration gradient, as well as both endo and exocytosis, because we're moving lots of things at once, or very, very large things at once, and pinching off a piece of that cell membrane of the phospholipid bilayer. The things that do not require energy are those passive transports, things that do not require energy because they go with the concentration gradient. So just plain old simple diffusion straight through the cell membrane, or facilitated diffusion going from high to low concentration through a protein channel. That was cell transport. I hope it helped.